Today we will discuss about how the cell experience mechanics from the ECM. We have discussed many times in our previous videos that whenever the cell experience the stiffer ECM, they will activate certain cellular pathways such as ROC pathways, ROA, MLCK, and FUC pathways, and that will helps in changing the this cellular that will help them in this change that will help them towards handling the stiffer extracellular matrix environment. And so in this case we will discuss how those stiffer ECM will cause those changes, and how they regulate the gene expressions. So on the left hand side, this is about the stiffer ECMs changes that are occurred within the cells, and on the left hand side it's about soft ECM changes that are occurring within the cells. So when the ECM is stiff, and so what will happen is these focal adhesions bonds are formed, and because of this affecting formations are also strengthened through this feedback loop pathways, and these feedback loops pathways are being triggered as soon as these focal adhesions are made a strong bond with the stiffer ECM, and that will trigger these high traction forces that leads to this feedback loops activations, such as these ROC pathways. ROA will helps in triggering these ROC pathways, and that will helps in upregulating MLCK and FUC pathways, and that helps in strengthening these focal adhesions. So in return, what will happen to that is that this affecting formations will go into the affecting formations will affect the nucleus gene expression data, or it will lead to the change in the it will lead to the epigenetic changes occurring within the cells in such a way that it will either promote uh, it will majorly promote these EMT pathways. Whereas what will happen when there is a soft ECM, there is a weak dynamic force occurs, or the weak interactions occurs between effectins and the extracellular matrix, and that will lead to this weak feedback loop activations, and thereby the effectins formations are not that strong as the traction forces are low, and these things get triggered when the traction forces are crossed beyond a partial threshold level, and if the traction forces are a below a threshold levels, then this weak dynamic force get triggered, and because of this the epigenetic changes that are occurred through this soft ECM interactions are low compared to this stiff ECM interactions. And this and these are the some of the factors or components that are related to this focal adhesions interactions. So in the case of stiff ECM, these interactions are very high, and in the case of soft ECM, these interactions are very weak and dynamic, based on this dynamic based on the strength of, or based on the interactions of this uh, integrins to this soft ECM, and this will cause, and if the because of the weak dynamic force, there is a disassemble and assembles of this effectins occurs more frequently in the case of these cells that are surrounded by the soft ECM, compared to the case where cells are surrounded by the stiffer ECM, and the change in this gene expression will cause this factor translocation from the nucleus into the cytosols of the cells, and that will we can say um, aggravate the conditions that are present within the cells. That is, if they are beyond the partial threshold level, then it will promote these feedback loops, or it will promote the strengthening of this affecting, and if they are in the weak dynamic range, then it will suppress. Or they are in weak dynamic range, then these translocation factors will work accordingly. That is, it will either help in this assembly or disassemblies, or it will suppress or upregulate this feedback loop based on the forces that are being received from the soft ECM interactions to these integrins, and that leads to this the strength of focal adhesions. So I hope you got a basic idea of about how this cell experience mechanics from the ECM. And if you need to understand more about this mechanics in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.